To manufacture modern therapeutics, biologists encounter a fundamental biological barrier: the programmed lifespan of highly specialized cells. Our immune systems can generate incredibly specific defense molecules, but the cells that produce them are naturally mortal. Take plasma B cells. They act as efficient biological factories, capable of pumping out perfectly targeted antibodies. However, their cellular clock is limited to a lifespan of mere days to weeks. On the other end of the biological spectrum are myeloma cells. These are cancerous plasma cells that have achieved cellular immortality, capable of dividing in a laboratory culture indefinitely. This immortality is medically useless on its own. Because they are malignant, these myeloma cells do not produce the functional antigen-specific antibodies required for therapy. To manufacture clinical-grade targeted therapies at scale, we must force these two disparate cell types to combine their distinct biological advantages. This is the core of Hybridoma Technology. It utilizes cellular complementation, physically fusing a mortal B cell with an immortal myeloma cell to create a permanent, single-specificity antibody factory. This diagram illustrates the hybridoma pipeline. Executing this requires a three-stage engineering sequence: inducing cellular fusion, applying a metabolic filter to isolate the target hybridoma, and performing high-resolution purification. The pipeline begins in a living host. We inject a target antigen and an adjuvant into a mouse, triggering the immune system to expand the specific B-cell clones that recognize that antigen. We then isolate the spleen, which provides a dense suspension of these activated B-cells. At this stage, they are productive but still mortal. To this suspension, we add a specialized line of myeloma cells, such as SP2/0. These lines are engineered to be non-secreting, ensuring they don't produce any competing, non-functional antibodies. With both populations mixed in a culture, we introduce the chemical catalyst polyethylene glycol, or PEG. PEG dehydrates the space between the cells. Removing the surrounding water forces the cellular membranes into such extreme proximity that the lipid bilayers begin destabilizing. This forced destabilization allows the membranes to merge. The resulting fused cells pool their genetic material, eventually stabilizing into true hybridomas. A beat of caution: however, PEG is non-selective. The resulting culture is a mixture of unfused cells, useless B-to-B fusions, and the rare functional hybridomas that must now be isolated. The next challenge is separation. We must find the single desired hybridoma among millions of unwanted cells without manual sorting. Cells typically use two distinct routes for DNA synthesis. The primary route is the de novo pathway, while the backup is the salvage pathway. We can control these routes using HAT medium, a mixture of hypoxanthine, aminopterin, and thymidine. The aminopterin in the medium inhibits the enzyme dihydrofolate reductase. This action effectively shuts down the de novo pathway, preventing the cell from synthesizing nucleotides through its primary route. With the primary route blocked, survival depends entirely on the salvage pathway. This backup route requires the HGPRT enzyme to function. Now, look at the cells in our mixture. The original myeloma line was chosen because it is genetically deficient in the HGPRT enzyme. Without HGPRT, these cells cannot use the salvage pathway. Because HAT medium blocks their only other option, the unfused myeloma cells starve and die. The unfused B cells do possess the HGPRT enzyme, so they can use the salvage pathway to bypass the chemical blockade. They survive the HAT medium initially, but because they lack the capacity for infinite division, they naturally reach the end of their lifespan and expire. That leaves the hybridoma. It inherits the HGPRT enzyme from the B cell and the trait for infinite division from the myeloma. Because it has both, the hybridoma survives the HAT medium using the salvage pathway and continues to divide indefinitely. This metabolic filter ensures that the survival of only the hybridoma is dictated by the presence of the necessary metabolic machinery. Surviving the filter only proves the cell is a hybridoma. We still have to verify that it is producing the specific antibody we designed it for. We screen the surviving colonies using ELISA or flow cytometry. These tests confirm which hybridomas bind correctly to the target antigen. To ensure every antibody produced is identical, we use limiting dilution. We dilute the suspension across a plate until exactly one single cell is isolated in its own well. When that single cell multiplies, every antibody harvested from that colony is a clone of the original parent, ensuring true monoclonality. Once this single specific clone is isolated, the process moves into industrial-scale manufacturing. Modern production has moved away from mouse-based harvest methods toward in vitro standards. We now use GMP-compliant bioreactors to grow these hybridomas in controlled environments. The raw fluid from these reactors is a complex mixture. We begin purification with clarification, using centrifugation to remove the bulk cell mass. We then perform primary isolation through ammonium sulfate precipitation, which relies on the salting out principle to isolate proteins based on their solubility. The most selective stage is affinity chromatography. The column is lined with protein A and G, molecules that have a precise binding affinity for the antibody. These proteins lock on to the Fc region of the IgG antibodies. This allows us to anchor the product while cellular debris, DNA, and stray proteins flush away through the bottom of the column. After the antibodies are released from the column, we use ion exchange chromatography, sorting them by net charge, and applying viral filtration. This ensures absolute safety. This sequence of biological and chemical engineering is what transforms a cellular fusion into a stable, manufacturable medical product. Early monoclonal antibodies faced a significant clinical hurdle. Because they were derived from mouse cells, the antibodies themselves were entirely murine. When injected into human patients, the body recognized these mouse proteins as foreign. This triggered the HAMA reaction, where the patient's own immune system would clear the drug, often causing severe side effects. To solve this, scientists used recombinant DNA technology to replace mouse protein sequences with human ones. This diagram shows the progression from murine to fully human antibodies. By transitioning from chimeric to humanized models, we successfully reduced the amount of non-self protein the human immune system could detect. Solving the problem of immunogenicity allowed these molecules to transition from experimental diagnostics to drugs that can be safely tolerated by the human immune system across multiple doses. This hybridoma pipeline is the mechanism behind many of the targeted cancer treatments and immunotherapies in use today. For example, rituximab is engineered to seek out and bind to the CD20 antigen on the surface of malignant cells in B-cell lymphomas. Similarly, trastuzumab is designed to target HER2 receptors in certain types of aggressive breast cancers. While the pipeline is complex and resource-intensive, it provides us with the ability to produce infinite quantities of molecules with exact specificity. Hybridoma Technology is a study in cellular complementation. By taking the targeting capability of a mortal immune cell and merging it with the longevity of a malignant one, we created the tools for modern precision medicine.